Genetically modified organisms. Genetic engineering can be used to modify bacteria, viruses, animals, and plants. Transgenic organisms are created by genetic engineering techniques where a genetic sequence is transferred from one organism into another with the help of certain bacteria. The following video, linked here, describes the problem of trypanosomiasis, sleeping sickness, in sub-Saharan African cattle and how genetic technologies are being used to tackle the problem. Transgenic cow. A disease-resistant gene in an indigenous cow has been identified, along with its DNA sequence. The goal is to move the gene into the genome of a domesticated cow to improve its resistance. This link explains how to transfer a disease-resistant gene into a cow, creating a transgenic cow. The disease-resistant gene is cut out by restriction enzymes and then isolated using the process of electrophoresis. PCR is then used to make billions of copies of the gene. There are now enough pieces of the desired gene to move them into a plasmid. The gene copies are combined with opened plasmids and carry out millions of ligation reactions to join them, producing a mixture of plasmids with added genes, some useful and some not. The bacterial colony that carries the disease-resistant gene is placed in a large liquid media culture, greatly increasing the number of bacteria cells as they reproduce in the favorable conditions. The DNA needs to be transferred into cultured cow cells. The bacterial cells containing the gene are placed in a container with the cow cells. While it is a rare event, if enough copies of the bacterial cells are present, some cow cells will take up the introduced DNA. Once that occurs, the new gene will integrate into the cow cell's genome. These new cow cells are called transgenic cells. Transgenic animals. Using reproductive technology methods, fertilized embryos from mother cows that lack disease resistance are isolated. Using tiny needles and the process of microinjection, their nuclei are replaced with the nuclei from the cultured transgenic cow cells, creating transgenic embryos. In this diagram, an egg cell is fertilized by a sperm cell from a microinjector. The micromanipulator holds the egg in place. Reproductive technology methods like this one reduce the element of chance in reproduction. The transgenic embryo is then implanted into the mother cow's uterus. Her calf will be transgenic, disease-resistant, and will be able to pass that disease resistance gene to its offspring. The desired gene has been added to the gene pool of this population. The video found at the link below explains how modern breeding programs that combine genetic engineering and reproductive technology will improve the quality of cattle. Genetically modified crops. The genetic engineering approaches to making transgenic cows are similar to the methods used to make genetically modified crops, GM crops. Genes from other organisms can be introduced into crops to improve their nutrition content, make them resistant to pests, extreme temperatures, drought, and high salt, and make them less dependent on nitrogen fertilizer. One of the earliest GM crops developed in Africa is the insect-resistant cowpea, shown here. The cowpea is an example of a genetic modification success. By moving a gene from a bacterial species into cowpea, African scientists have produced crops that require less pesticide and give higher yields. Maruca vitrata is an insect that bores into the pods of cowpea, destroying the crop. A soil bacterium Bacillus thuringiensis releases a toxin that binds to insects' gut epithelial cells and kills them. Because animals lack the receptor for the toxin, it has no impact on humans, but it can kill Maruca vitrata. Genetic engineers have moved the gene encoding the toxin from bacteria into cowpea cells, producing plants that defend themselves against insect pests. GM agricultural products have resulted in increased productivity and improved nutrition for many people, helping to prevent starvation. But there are issues with GM crops that are discussed. Pollen spreads easily by wind, water, and animal movements, and may fertilize wild, related species, altering their course of evolution and disrupting ecosystems. Crops that are bred for size, early ripening, or shelf life may not have the same nutritional value or flavor as their wild ancestors. One of the most successful uses of genetic engineering for food has been the development of golden rice. 2 genes that synthesize beta-carotene that leads to vitamin A were inserted into rice, transforming it into golden rice. Regular rice has 0 vitamin A. Golden rice is critical in areas where vitamin A is in short supply in people's diets. Vitamin A shortage leads to vision problems that range from mild to very severe, including blindness, as well as diarrhea and measles in children. A new version, Golden Rice 2, produces 23 times as much vitamin A as the original. Golden rice has been approved for use in many countries, including the USA, Canada, Australia, and New Zealand, and approved for planting in Bangladesh. There are organizations that argue against the use of golden rice, as it is a GMO. There are drawbacks of making transgenic farm animals and genetically modified crops. It is time-consuming, expensive, and requires trained technicians and laboratories. Some transgenic animals and GM crops are not as healthy as their natural counterparts, and some complications cannot be foreseen. There is concern that the transgenes could spread into wild or indigenous populations, disrupting the normal course of evolution. Nevertheless, these techniques are being used all over the world to improve agricultural productivity.